If we were to look at a particular atom or atoms inside of a molecule or a fluorophore, what we're going to find is that these atoms have electrons, and these electrons have allowed energy levels. And what happens specifically with special kinds of fluorophores is we're able to excite these electrons to jump into different energy levels. And so if we were to consider a photon of a particular wavelength coming in and striking this electron here, what will happen is we are going to see this electron jump up into a new energy state and then relax down into its previous energy state. And so during this relaxation process is when we see these new wavelengths of photons come out of our sample, and specifically these wavelengths are going to be different. And this different wavelength of photon is something that's very interesting to us because it's basically telling us that a fluorophore is present inside of our sample. And so to spell this out a little bit more clearly, what we're going to see is that we have a lambda excitation as well as a lambda emission. And when we look at the photons that have excited a particular fluorophore, what we're going to see is that these photons are going to have a higher energy level than do the photons that come out of the sample when that electron is relaxing. And so what this tells us, and what we should know based on the Planck's constant law, is that the lambda excitation is going to be a smaller value than the lambda emission. And this is because energy is inversely proportional to the wavelength. So having a longer wavelength of a photon means that that photon has less energy. And so what this tells us when we look at a particular plot, and if we were to consider a fluorophore like Psi5, is that we're going to see that Psi5 has a particular band of photons that will excite it. And so when we are looking at this, we can now label this plot that I have in the bottom left here as an excitation band or range. I'll just call this an excitation band. And when these photons in this excitation band are absorbed by your sample, by your fluorophore specifically, your fluorophore is going to release, it's going to emit a photon of a lower energy that has a higher wavelength or a greater wavelength. And so this would be your emission band or EM band. And so in this emission band, this is where we can have detectors. And if we are seeing photons come out of our sample that are inside of this emission band, then we can conclude that these fluorophores must be present inside of your sample. And so the way that a flow cytometer works is we'll have a laser or some kind of light source like an LED. And the purpose of this light source is to basically generate the photons that are inside of an excitation band for your fluorophore. So if our cells here, and these cells are the little red dots, had the Psi5 fluorophore on them, and they were bombarded by photons that were within the excitation band, what happens next here is that photons in the emission band, and these photons would have literally a different wavelength, and photons are light, so these photons would have a different color. These photons are going to be emitted from your cells if they have the Psi5 fluorophore bound onto them, and when they do that, you have detectors downstream that are able to determine the intensity of the emitted photons, thus quantifying the presence of these Psi5 fluorophores on your cell population, because you're looking at individual cells one at a time, and these things are extremely fast. They've got very high throughput, and so very quickly you're able to determine how many cells in your population have a particular fluorophore present. And this is how the flow cytometer is working. And so oftentimes what you'll also see is that you're not just going to be using one fluorophore, you'll be using several types of fluorophores to perform something that's referred to as a multiplex assay. And in this multiplex assay, you'll have other fluorophores like Texas Red, which is sometimes called T-Red. You'll have DAPI, which is another channel. You'll have PE, as well as Psi5, and there are several others, but basically these are types of fluorophores, and so you can have each one of these detectors, one for T red, the other for Psi5, and so on. And so this will help you look at multiple proteins or multiple different aspects or qualities, characteristics of a cell population all in one go. And so this is what makes flow cytometry a very powerful tool for us to be able to very quickly analyze and define the characteristics of the population. A final thing that I wanted to talk about here is that when you see people talk about fluorophores, very commonly they'll tell you, oh, lambda excitation is equivalent to 350 nanometers. And even though they say 350 nanometers, and this seems like a single wavelength of light that will be emitted from a particular sample, and specifically I want to actually not look at the excitation, but I want to be looking at the absorption, or an absorption is excitation. And in this case, if we were to look at this plot and someone was to tell you this is 350 nanometers, I want to make it very clear here that per quantum mechanics and the lack of determinism at the quantum level, we cannot say definitively how a particular atom is going to absorb and emit photons. And so what we have to do is we have to define an absorption spectrum or a range. And so the reason we call this lambda equals 350 nanometers is because it has a peak absorbance at 350 nanometers. That doesn't mean that it is unable to absorb photons at different wavelengths of light. What we can see with this particular fluorophore is that it's able to absorb photons at around 330 nanometers, as well as photons at other types of wavelengths too. So you've got an entire range of photons that can be absorbed by a particular fluorophore. We're just reporting the peak absorption because it's an easier number that we can quickly toss around and share with people rather than a range. So it does not do it full justice, but it does help us refer to these fluorophores and determine whether or not they would fall within a particular excitation band. And so if I was given this particular fluorophore, if we were to call this a T-red fluorophore, and someone was to ask me, I have this particular excitation band, I have a light source that is able to produce photons within this particular range, and they said, can I use this in my assay with this flow cytometer? The answer to that would be no, because my excitation band does not make photons that are able to fall within this particular wavelength of light that is required by a different fluorophore channel. And so this is why we have different channels, which is how scientists and engineers are able to very quickly determine multiple characteristics and intensities of these cells using flow cytometry. So that's going to wrap things up for this video. I hope it helps. Let me know if you have any questions. Thank you all for watching, and I'll talk to you next time.